again, and we're back for Chapter 3, Part 2. More fun with cells. So now we're going to talk about where new cells come from. Because actually, that was part of a debate. I wonder if I could get away with showing you. It's my TED Ed. Well, anyway. There is I have a video online if you haven't looked it up. It's the history of the cell theory. I mentioned it in the last lecture as well. It's all about how we came to understand where cells come from and whatnot. And so it's fun. It's a fun little animated short that I got to do with TED Ed. The TED lectures and whatnot. They have an education wing. They try to put out like little videos, animated videos, kind of like it. Originally the logic was to kind of make it in the style of what is it, Schoolhouse Rock, except with less singing, because I can't sing. There was no way that was going to happen, me singing about the cell theory. But anyway, it's a fun story. But anyway, all right, so let's talk about where do cells come from. And this actually was interesting until we finally observed it under a microscope. For the longest time, we used to think that cells just used to crystallize into existence. It was something called free cell formation, where if you had enough oxygen, it would just go, there it is. And no. So anyway, so this is not how that worked. And then somebody under a microscope, who was actually a scientist named by the name of Robert Ramack, actually said, hey, I think I see these cells actually splitting into two. And he showed it to a friend of his, who then stole it for himself. Because that's the fun thing about science, is like, if you don't publish it quickly and get it under your name, you might get it stolen by somebody else who will publish it even faster. It's academia. We'll get into that later. There's also some stories that kind of fit into this as well. All right, so speaking of the cell cycle, with the exception of gametes, which is the eggs and the sperm, these guys, they don't go through mitosis. They go through double mitosis, which is called meiosis, and we'll talk about that later. Pretty much everything except for your neurons, which are too specialized to do this. So once you've made, so many neurons, that's it. That's all she wrote. That's why we can't get them back if we destroy them or kill them, because these guys cannot go through mitosis. Maybe hopefully one day we can figure out some way to replenish them, but right now, once you kill a neuron, it's dead. It's not coming back. However, other cells, on the other hand, have a problem. There comes a point in a cell's life where it gets too big, that its cell membrane cannot hold everything inside. There's a mathematical principle behind this, and I get into it more in biology, but I'm not going to really do it here. Just know there comes a point where a cell has to choose one of two options. Die, which is called apoptosis or apoptosis, or split into two new cells. Because it's just gotten too big. Because if it continues, it will die. It will pop its own cell membrane, and what's inside will come out, and yeah, that's not good. Now, there is a third step to that called cancer. And that's where the cell decides to just go crazy. But we'll talk about that in a minute. So anyway, I have a really good Bozeman video on it. I would highly recommend watching, and I'll say that again later when I hit it in these notes. Anyway, so the life cycle of a cell, it's also known as interphase, and then when it decides to split, when it goes through the splitting, is mitosis, and then cytokinesis. Cyto means cell, and kine means to move, which is why it's called kinesthetics, for exercising and whatnot. It's moving. So anyway, it's kind of funny. Interphase is also known as the resting stage, but in reality, the cell's actually doing its job. So what happens is there comes a point where the cell, so here's a baby cell, and by baby I mean it's just smaller. It's not like baby. So it's a smaller new cell, and it goes through G1 in the life cycle. So this is the life cycle of the cell. And that G1, it's doing its job. It's in interphase. He's doing his job. He's if he's a heart muscle cell or a skin cell, we're constantly sloughing those off and making new ones of those. So G1, he's doing his job, he's growing, and he hits this point called R. And he's so big at the point where, like I said, his cell membrane's stretched really thin because the inside is growing more rapidly than the outside can grow. Like when you get too much rain on your tomatoes and it makes the outside of your tomatoes split open. I've hit that. If you grow things. So yeah, that like, because there's too much water on the inside and it puts pressure on the skin and it breaks. And there you get a split in your tomato, and then you're like, no, there's a split on my tomato. And so it's basically same thing with cells. So it gets to a point where the inside cannot be held back any longer by the cell membrane. So he has a decision to make. He's sitting there, well, I can pop and die, which actually is a viable thing. There are cells in our body that are pre-programmed to die. We'll talk about that in a minute in that process as well. Or he could sit there and go, okay, there's a space open next to me. I'll just split into 2 new cells, and then we'll start again smaller and newer and completely identical. And that's what a lot of cells choose to do, especially skin cells. We're constantly doing this with skin cells. So imagine we're talking about skin cells today. Actually, we'll be talking about skin cells even more in chapter 5 and 6, so it's coming. But there's also a third place where he can choose, and that's if his DNA is really, 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 really damaged. And unfortunately, that's cancer. This is where cancer comes from. It is a disruption of the cell cycle. And it's where, unfortunately, the cell chooses the third path, which is just complete and utter anarchy. It's like, I don't need your controls. You can't control me. And then, unfortunately, he goes pretty much crazy. He starts just growing abnormally. He won't, go through apoptosis and self-destruct. He just keeps growing abnormally, and that's not good, because cells that do their own thing put into jeopardy the whole organization of our body, and therefore our body is like, stop that, and the cancer cell is like, no, and now I'm going to recruit friends to do the same thing, because there's a third option that nobody knew about, and you didn't tell anybody about. And it's like, yeah, we didn't tell you because we like living. So cancer is like when the cell cycle goes wrong. Very, very, very wrong. So if it chooses to die, then it stops right about here. If it chooses to go ahead and go into replication, or go ahead and go into mitosis, it then enters the S phase. The S phase is when it's starting to replicate all its DNA and gather enough energy to go ahead and go through the process, because this is a very energy consuming process. So S is where he's gathering energy, and replicating his DNA. So he's making a copy of everything. And then from there, what he does is he goes into G2. When he has enough energy and he's replicated all his DNA, he does a check. So G2 is where he's double checking everything. Do I have enough energy to go? It's kind of like that right before they do a launch, where they go to the people in the control command center and they go, no, go. And, each group that, is in, over the control of major systems of the rocket ship goes, go, or no go. Because if there's one no-go, then the mission, and then the launch is scrubbed. But pretty much, when the person who's in command of the operation stands up and goes, go, no, go, and then they all sit there and say, go, go, go. It's just like that, except it's yourself. So I wonder if it's just as dramatic, too. Anyway, who knows? Anyway, so the G2 is just like that. It's double checking everything. Do I have enough energy to go forward? Have I replicated all the DNA? Did I replicate all the DNA correctly? And it's not like hoogly-boogly? Because that can happen too. We'll get into what happens when the DNA doesn't replicate correctly. That comes later. And so after it goes through the checks, it then goes the give ahead and goes straight into cell division. So that's where we go through mitosis, and we get 2 new identical daughter cells. So resting stage is G1, the decision is R, S is where it's building up, enough energy and replicating the DNA to make sure it can do everything and replicating, organelles and whatnot. And then again, the G2 is where it sits there and goes, all right, double check time. Do we have everything we need? Is everything good? We all go for splitting. And then M is mitosis, where it splits into 2 new identical daughter cells. So what about mitosis? What about it? So this is just a quick, overview of the steps that's going on. Technically, there's 4 main steps. There's prophase, pro for first. Then there's metaphase, where it lines up in the middle. That's how I remember it. Meta actually means changing and stuff, or whatever the heck Facebook's company has become, parent company. It's now Meta, whatever. Metaphase. It's better than meta. And then anaphase, ana means apart, so it's pulling it apart. And then telophase is where it completes. Telophase, it means far and or complete, so it splits into 2 new identical cells. So basically what happens is, again, like I said, if we're sitting here and we're going into S, it's replicating its DNA. That's what's happening right here. All these guys coil up, makes an exact duplication of itself, which is how we get these chromosomes. So these are our friends, the chromosomes, those X-looking things. Now, chromosomes are made up of 2 things called chromatids. 1, 2. So each one's buttoned together by a centromere in the middle. But anyway, so what happens is now that's when, remember when I said the delicious looking centrioles, or the things I said that look like, I just said it a minute ago. Churros. Split. So they've been a pair for a very long time, just hanging out while the cells have been doing this main job. Now, it's time for him to wake up and start doing his job. So what these 2 do is they actually start splitting and going to opposite sides of the cell. And they actually start shooting out these things called spindle fibers. So they push off each other and go to opposite ends of the cell. And what happens is these guys line up. There's actually motor proteins, like those little guys with the feet I showed you in the last lecture. Move them to the middle. And then what happens is they shoot out these things called spindle fibers. And the spindle fibers go down and attach to the centromere, both sides. And then they start pulling. So I liken it to fishing. So basically these guys, you line your ducks up and you shoot your lines out, and then it pulls them apart. So here we go. They pull them apart. Oh, by the way, they line up on I love how they call it, the cellular equator. It's just the middle. So, because, again, biology, we get really futzy with our terms, which is why physicists sit there and look at us like, you know what we call it a black hole? We call it a black hole because it's a black hole. And I'm like, yeah, what for you? No imagination. Should have named it Bob. Anyway, moving on. So they pulled it apart. So anaphase, this is anaphase. So the spindle fibers have now pulled these apart into separated chromatids, and they're reeling it into each side. And so they've reeled it into each side. The cell out the outside starts to pinch off. This is the beginning of telophase. And 2 new nuclei start to reform as it pinches off. And this is called cytokinesis, where the cytoplasm is dividing and pinching off. And then, bada bing, you have 2, and the DNA relaxes back into its relaxy form, which is this form right here. Although it doesn't have eyes, but still. The double helix form that we know and love. Anyway, turns into 2 brand new, identical daughter cells. And we do this constantly. We're doing this all day, every day, sloughing off our skin cells. It's actually why about 98% of all dust in your house is actually your own skin cells. So yeah, if you don't like dusting your house, you only have yourself to blame. Sorry, science. Again, if you want to see it in action, which, like I said, videos are worth everything, highly recommend you watch our friend Hank go through mitosis, which is really, really good. I like using this video because it's just a good recall if you don't remember the steps of mitosis from biology. So, if you need refreshment, here you are. Also, like I said earlier, he does a really good one. I have one on how cancer cells happen, which is really important for later on because we do pop up. We do talk about cancer on and off throughout this entire course. We don't have one entire chapter dedicated to cancer. It's just on and off, on and off, on and off. So it helps to know why cancer happens and how cancer happens. Just on a general thing, remember everybody's cancer is completely different. Even if two people have, the same lung cancer, like unfortunately my father died of lung cancer. Of course, then again, he had been smoking since he was 10 because he worked for a cigarette company and they paid him in cigarettes. Anyway, so long story short, his lungs were not so hot. So long story short, with cheese, like two different people can have, even if it's classified as the same, it can be completely different because of their own unique biologies. Which is why I get mad when somebody's sitting there going, the government has the cure for cancer, it's just they don't want to give it to us. And it's like, why? Because there is no one cure for cancer. It's impossible because even like I said, between two people, even if they had the exact same cancer, it'd be different because of their biologies. Which is why we're looking at DNA tailored cancer treatments in the future, which I think is exciting. But anyway, long story short, sorry, I'll get off my horse or whatever. I will get off my large animal. So highly recommend this video, especially since we're going to be talking on and off about cancer here and there throughout this entire course. So anyway, so cells that do not follow mitosis, there are several hundred specialized cells, and the process of specialization is called differentiation. And we get that through our friend, and you've probably heard of him, the stem cell. This is the plushy version. He's different colors because he can be anything. That's why he looks all tie-dye-y, I guess. Anyway. But anyway, the stem cell. So the stem cell is one of the few cells that can choose his path in life. He can become anything in your body, and you've probably heard this already. Just, at passing with stem cells. So they can undergo self-renewal, they can make more of themselves, or given a signal, they can choose to turn into almost anything in your body. Now, the further more it goes, the more specialized it becomes, it can't go backwards. So remember, stem cells, once they choose a path and they go down that path, they cannot go backwards. That's the thing. That's why it's like trying to find a good place of harvesting stem cells is rough. Because we're getting pretty good at possibly using stem cells inside of umbilical cords, which is why there's a whole thing about storing umbilical. There's a whole thing where you can send your baby's umbilical cord to an umbilical cord bank and they'll freeze it and keep it for you for, paying out the nose for it. But that could be used later to, help with certain diseases, which unfortunately, I couldn't do with my son. It just wasn't feasible at the time. Anyway, and you can't just put it in your freezer. It doesn't work like that. Anyway, so stem cells. Now, the next step under a stem cell, so if he chooses a path, so say he wants to turn into a red blood cell. So if he chooses a path, the next path, he becomes more specialized. He becomes a progenitor cell. And that becomes, he has now a more limited path of what he can choose because he's chose to specialize. Now he can go into one of the, I was just doing this with anatomy and physiology too. Not the lymphatic, that's going for immune system. The, I'm coming. Mitotic cells, that's where you get the red blood cells. So he becomes a mitotic progenitor cell because he's chosen a path, and then he has to choose another path to get more specialized and more specialized and more specialized on down the road. So, for instance, the things that we like to talk about that you hear in the news all the time are the totipotent embryonic stem cell. These are the ones that can turn into pretty much anything in the body. So this is the things we want to harvest because then we could trigger it and hopefully grow a new liver, say you need a liver transplant, or a new kidney, say you need a new kidney transplant. Wouldn't it be nice to grow it from your own stem cells? Except us adults, we don't have many stem cells because we're already adults. They've already all chosen their pathways. So we have to seek our stem cells from other places. Now, if they choose to instead, like I said, they can make more stem cells like themselves, or they can choose a path. Now, one of the main paths are basically how our things started when we're growing. It can choose to go through the endoderm line, the mesoderm line, or the ectoderm line. Endo means inner, meso means middle, ecto means outside. So if you chose the ectoderm line, you get to turn into a multipotent stem cell, which basically means you can turn into anything between skin or neurons, because your ectoderm line turns into those things. Mesoderm line, again, he can choose to go down and turn into a multipotent stem cell, but he can only make a red blood cell or heart muscles or something like that you find, Again, these guys can't turn into these guys. Oops, gone too far. So these, once they've chosen their line, it's kind of like, choosing your team, I come from a long line of Yankee fans. I mean, granted, there could be a Mets fan among the family. I don't know where, but there could be. But, my family chose the Yankees back, and gosh darn it, we're all going to choose the Yankees. Now, granted, we're not cells, and we can choose later. We could have that one wild person going, yeah, I like Boston. And then they're like, oh, Sean. No, we don't do that. We just make fun of them. Anyway, so same thing here. It chooses, and it can't unchoose. It's only ways this way. It can't go over and be like, oh, I don't want to be a red blood cell. I want to be a pancreas. No, it doesn't get that. So it's like, once you choose, that's it. It's like houses in Harry Potter, I guess. I don't know. Except it's not a sorting hat. It's cells. So, hopefully that makes sense. Now, granted, there are places where we can make them go backwards a bit, but we can only make them go backwards into their line. So, like, for instance, if one's being turned down to a neuron, we could take the bone marrow, the skin, cord, blood, and deciduous teeth and actually make it go back to being an ectoderm line cell so it can choose another path. But we can't go back to that totipotent embryonic stem cell. So, yeah, there are ways to induce it to be going back to a pluripotent, but it cannot go back, at least at our technology right now, to a totipotent. Does that make sense? I hope that makes sense. Now, this is where I get on my rant. Actually was a rant that I inherited from my biology and anatomy professor. So, and that is apoptosis or apoptosis. And you notice, hopefully coming up to this, I've been saying it funny, or at least two ways, and there's a reason for that. So, apoptosis or apoptosis, which is what you're going to hear the majority of people say, apoptosis, and there's a reason for that, is programmed cell death. Now, there comes a point where some cells just are not doing anything and they have to die off. Interestingly enough, when we're born, we actually have to do this with our own nervous system and our brain. We've actually made way too many brain cells, which is a good thing. You always want to have more than less, you know what I mean? So we made way too many. So they're kind of like, redundant. So we start telling them, hey, die, but in a nicer way. Because you're redundant, we don't need as many as you if we thought we did. So they start going through programmed cell death or apoptosis. Now, it's also, like I said, if you find older school people, they'll say apoptosis. What happened is, once upon a time, there was a scientist who wrote the book on this, literally. He got a Nobel Prize for figuring out how apoptosis works. And how it was different from necrosis, which is where a cell dies of unnatural causes. And usually that triggers the immune system to go, whoa. This won't trigger the immune system. This will trigger the immune system. Necrosis, which is just, unnatural cell death. Something killed it, like a virus, or, or it's being cancerous and weird. And like the immune system goes. So anyway. So he figured out the difference, and he wrote a paper, and he got the Nobel Prize for this, which, if you get the Nobel Prize, it really kind of mean. Cements you as being the ultimate authority on this subject. However, every time everybody heard him speak, he kept calling it apoptosis, not apoptosis. He really kept emphasizing the pop, and it drove my professor out of his freaking gourd, because before that, everybody called it apoptosis. And he's just like, he'd go on this 10-minute rant, and it was so funny. So funny. In the middle of class, just going, it's apoptosis, not apoptosis. So that's my apoptosis, not apoptosis story, because if you get the Nobel Prize in something and you start saying one of the terms weird, guess what? The entirety of science will fall in line behind you and say that term weird too. So that's why you'll see a lot of videos talking about apoptosis, not apoptosis. So yeah, potato, it's so that, but I mean, it's just funny. So I just want to more for you. But anyway, like I said, normal cell, the membrane will start blebbing. Basically what it does is it releases all, if it's, remember what I was talking about, the lysosomes, it pops its lysosomes, and it condenses, and then it fragments, and then it turns into apoptotic bodies. And these apoptotic bodies are usually gobbled up by a white blood cell. However, necrosis is when the cell, something happens to it, like, a viral attack or a bacterial attack or something like that, or even poison. Necrosis just outright kills the cell when it wasn't programmed to die, and that's why it's called necrosis. So apoptosis, it was supposed to die. Necrosis, it probably wasn't supposed to die. That's the difference between the two. One was programmed, and hey, we don't need you, you're redundant. Please go pop yourself. Whereas necrosis is like, I'm being attacked, help, help, help. And they actually, because they die in that way, they actually put out chemicals that actually attract immune system cells to them. So as they die, they even kind of leave a note going behind them going, I've been killed, here is what my killer looks like. And the immune system comes over and actually checks it out. We'll talk more about that when we talk about the immune system next semester. So anyway, I know I'm mentioning so many things that we talk about later, but anyway, just fun stuff. All right, there you go. That is the difference between, that's a cell cycle, mitosis, cancer in a nutshell, how stem cells work, and apoptosis or apoptosis and necrosis. And next one we're going to get into the nitty-gritty of how mitochondria work and other fun things like that. Like I said, I'm going to kind of like, again, this is drive-by sciencing. So I don't blame you if you're sitting there going, trust me, I get it. So with that said, I'll see you in the next lecture. Bye. Say bye, Monty.
Bye-bye.